Hello YouTube! Today I'm going to be explaining transcription in eukaryotes. Transcription is the process that DNA uses to copy the template strand of DNA into RNA. Now, there's 3 steps in this. The first one is initiation, then we have elongation and termination. Initiation, the transcription is initiated by RNA polymerase holoenzyme. The holoenzyme is the sigma factor and RNA polymerase combined. The core enzyme binds to a specific sequence on the template DNA strand called the promoter. So RNA polymerase just wanders down the DNA double helix until it finds these 2 promoter regions, one which we call the -35 region, which is TTG ACG in case of E. coli, and the -10 box, which is TAT AAT. It binds to these 2 regions, and it starts transcription at the +1 site. As soon as it binds to this, it will open up from about to +3 to -11. And then we go to elongation. Elongation it starts after it's unwound. It unwinds in front with the direction of transcription, and it rewinds it after it is done with this particular section. So it starts by using RNTPs, putting them slowly, getting them one by one, and slowly assembling them into the RNA with opposite binding proteins. So after elongation, after the RNA has been made for a certain time, it will reach a point which is called a termination. Termination is initiated by a specific sequence, in this case, that repeats itself after, in this case it was here 8 base pairs. It repeated the exact opposite. So here C bound to G, G bound to C, C bound to G, C binded to G, G binded to C, and C binded to G. So as it copied the first and the second section later, these were free-floating, and then they came together because they're exact opposites and combined together. This formed this hairpin formation. After this hairpin formation is formed, it replicates the 7A tail into 7 uracils in this case. And it always does this from the 5' to the 3' direction. This here is the RNA, while this here is the template strand. After this has been done, the RNA polymerase releases and it stops transcription. Now, in case where we have this once again, and the -10 region would not exist, that would mean RNA polymerase could not initiate transcription, and so no protein that could synthesize sugar would be made. Thank you.